So I'm very excited to have David Mazzo here, and there are multiple reasons for that. Having worked for NeoStem and having been very close with Robin Smith, the CEO, this is the first time that I'm not introducing Robin Smith. That's very, very interesting because it points to a lot of changes at NeoStem as a company. That Robin is still involved. She's still the executive chairman, if I get her title correct. But clearly the company is transitioning kind of away from the early stage roots where Robin pioneered and originated the company towards more of a traditional biotechnology company with a phase 3, with a phase 2, with a phase 1 asset, and really a very diverse product portfolio. My bias at NeoStem is I really love the Amersite product, and I'm very excited by that product. But what I've had to do as an analyst is I've really had to retrain myself to look at NeoStem very, very differently. I've had to look at it with a phase 3 oncology asset that can potentially change the paradigm of treating melanoma, as well as the phase 2 asset. And of course, the hottest area in biotech today is really when we talk about the ability to manipulate and change T cells. And so I think you're going to see that there's actually an early stage T cell asset here that could be very exciting. So with that, I'm excited to introduce David Mazzo.
Jason, thank you. Testing? Okay. Thank you, Jason, and thank you all for coming today and for your attention. I think Jason actually did my entire presentation for me, so I appreciate that very much. But I do want to take a few moments and give you an idea of the metamorphosis that has occurred at NeoStem over the last several months, beginning with some of the decisions that Robin Smith took last year and culminating now in a repositioning of the company and its pipeline. So, of course, we're a public company, so I will be making some forward-looking statements, and I'll ask you to keep that in mind. Like most of the companies here, our focus is transforming cells into therapies. But there are two things that I think distinguish NeoStem from many other cell therapy or immunotherapy companies. One of them is the operating subsidiary that we call PCT. PCT is an industry-recognized premier single-source provider of services to the cell therapy industry. It's a group that we consider a center of excellence for process development, engineering, clinical and commercial manufacturing of cell and immunotherapies. This gives us a real competitive advantage over most other immunotherapy companies who actually come to us as clients to provide these services. Not only is this group, which has been in existence for more than 15 years, been exposed to a diversity of problems and their solutions, which then they can bring to bear on the new problems posed by clients, but it also gives us the ability to develop our own products more effectively, more rapidly, more efficiently. And this is just a simple sampling of the more than 100 products, or rather clients, that PCT services. And you can see many of the very popular names in immunotherapy and cell therapy are on this list. And oftentimes people don't recognize that it's actually NeoStem through PCT that provides the services and the process development and manufacturing that allows these companies to move forward. The other thing that I think differentiates NeoStem, and Jason touched on this very nicely for me, is the fact that we are a company with a balanced and, I think, robust pipeline. Now, on the surface, some may look at this and say, well, you're in immuno-oncology, you're doing ischemic repair, you're in immunomodulation. That's a lot of different things for a small company. But I like to liken our pipeline to a grove of aspen trees. I don't know if many of you are skiers or if you've been out west very much, but if you watch the Discovery Channel, for instance, you know that one of the great examples of a symbiotic relationship in the plant kingdom is the aspen tree. On the surface, a grove of aspens looks like all individual trees, but actually underneath, there's a common root system. And so that's how you should look at our pipeline. We have individual trees, but underneath, there's a common root system. These are all products based on autologous therapies, and they're based upon the expertise in processing and efficiency that PCT brings to our pipeline. So, as a summary, we are a company that now has a management team that has many years in biotechnology, biopharmaceutical, successful international product development, and many of our scientific leaders are really world-renowned in their therapeutic areas. We have this set of platforms that are proprietary, that they provide a diverse yet balanced pipeline with a focus in each of the areas that mitigates risks for our investors but provides multiple shots on goal for upside. And we have this internal center of excellence that really positions us, I think, to be something much different, and it also provides a source of revenue for the company. So with that, I'll turn it over to Jason, and we can have a discussion.
So let's talk a little bit about the products themselves. When we talk about the melanoma oncology product, we're talking about a dendritic-based vector for the treatment of melanoma. How should I think about this when I think about Dendreon and Provenge? I get a little bit scared about the cost of autologous manufacturing, and I happen to be very aware that PCT was the clinical manufacturer of Provenge, and that Dendreon made a decision to go away from PCT. It's pretty clear in hindsight that that was a bad decision. Help me understand how this product is different than Provenge.
Yeah, I think it's different in several ways. One of the lessons that we learned from the Dendreon experience is that it's important to be able to keep the processing simple, not only for the patient's sake, but for the logistics involved in moving materials back and forth. As you know, when you're dealing with an autologous therapy, you're often taking source materials from a patient, bringing it to a processing center, and then returning therapeutic materials back to that patient. Another aspect that we learned is, whenever possible, to facilitate the logistics involved in that manipulation, it's best to have products that are stable and can be preserved, right? And so Provenge really had a very tight time window in which it needed to be processed and returned. It was a complicated process, and we learned as the process evolved out of our hands and into the hands of the originator that their ability to reproduce the work that was done at PCT with the same levels of efficiency and reliability were diminished, and that added also to their cost of goods. And so we have a situation in our program where we have a much simpler process, we have the ability to cryopreserve along the way, and we have, I think, a very clear understanding of the patient population and how it will be treated and where it will be found, and all of those things coupled with a 2- to 3-fold minimum improvement in processing efficiency over what was reported for Provenge gives us the sense that our cost of goods will be much, much more reasonable.
And let's just talk a little bit about phase 2 efficacy. One of the things that always struck me about Provenge is that the efficacy, I thought, was incremental at best. How would you describe the efficacy from the phase 2 data that we've seen here?
Well, I think you're right. And certainly in pharmaceutical development, oftentimes with small molecules, what we're looking for are continual incremental improvements in therapy. But here, I think the challenge and also the allure of cell therapy is that you can get a quantum leap. You can get a real step improvement. And that's the approach that we're taking with NBS 20, our melanoma program. The fact is that we can see, based upon our phase 2 data that was done 2 separate studies, one in a single-arm study and one an active control study, we saw improvements in overall survival that varied between 3 and 5 times those of the very best data reported by the current therapies, as well as those considered promising therapies in development. So we really see this as a paradigm-changing opportunity. To add to that, we're taking a completely different mechanistic approach here. So that does not exclude our therapy from being used concomitantly with some of the other anti-PD-1 or checkpoint inhibitor type therapies, which actually provides even a further opportunity for not only additive efficacy, but potentially even synergistic efficacy.
So is it fair to characterize this as the lead program at NeoStem, and what is the plan going forward in terms of the pivotal trial?
Well, it's difficult when you have a pipeline that has so many interesting things to actually say that this one is the priority. But to be fair, this is our lead program. It is the most advanced clinically. We're effectively a phase 3 company with a phase 3 asset, and that is the near-term focus for execution. The reason I can say that as well is, and we'll get to the NBS 10 program in just a second, our cardiovascular program, is that that program is at the last stages of phase 2. And so we have a program that's sort of stepping into the pivotal study, another program that's on deck, if you will, and then yet a third program in immunomodulation that is in the dugout and waiting to come up to bat.
So one of the challenges that the bears will try to throw at a program like this is that, do you really need a dendritic vector to deliver this efficacy? And isn't that 5 years ago thinking? How do you address that?
Well, it's interesting because one of the studies we did in phase 2, and maybe I should take a step back and very briefly describe the product. So the product is derived from first taking a tumor sample from a patient, extracting the tumor-initiating cells, expanding those cells, irradiating them, and then using that as the antigenic fingerprint for the cancer of that particular patient. On the other side of the therapy, there's a single leukapheresis that allows us to isolate the monocytes and ultimately the dendritic cells from that same patient. And when they're mixed together, that provides the immunotherapy. One of the things that we've seen is that the dendritic cells by themselves or the tumor-irradiated cells by themselves produced either no response or only a very modest response. But the ex vivo mixing of those two and then the subsequent administration produced the very dramatic response that we've seen, and that's the compelling reason for going forward with this therapy.
Okay, let's switch gears and talk about the cardiovascular product. One of the things that's been very frustrating for me as an analyst is, I look at phase 2 as information, as an informative design. And somewhere along the lines in the last decade, phase 2 became almost a pivotal-like trial, and we stopped looking for information, and we demand p-values on primary endpoints. For me, I looked at the phase 2 data at things like mortality, cardiac MACE, and hospitalizations as good data. Help me understand, what was the 6-month data? What was the 12-month data that you just reported at AHA, I think just a week ago, March 15th?
That's right. So, to take a step back, first of all, I completely agree. I've been doing clinical development and pharmaceutical development for almost 35 years, and there is always this tendency to want to jump ahead, right? Especially when you start to see really compelling responses in patients, and we're all motivated by seeing patients get well. But the fact is, as you pointed out, Jason, that phase 2 is a necessary informative step in the preparation of your pivotal trials. And in phase 2, you typically do things like look at exploratory endpoints to determine what might be the fastest route to a regulatory approval. You look at dosing, you look at patient populations, you even explore indication, and you get some operational information about the centers that enroll and that don't enroll. You look at how in the real world your compound and its administration really works, and that's exactly what the PRESERVE AMI trial was designed to do. Now, somewhere along the line, somebody decided that the primary, or one of the primary endpoints, which was an exploratory endpoint that looked at perfusion based upon an imaging technique, was the end-all and be-all, and that somehow that was going to indicate the developability of the product going forward. And in fact, all that did was inform us on whether or not that particular surrogate would be worthy of a discussion with regulatory bodies, because clearly the current guidances on cell therapy and cardiovascular medicine state that MACE and mortality, and in some cases ejection fraction, are the required primary endpoints. And those are the things that I suggest, as you pointed out, that people focus on, because the data for 6-month and 12-month interim in an overall 3-year follow-up in these AMI patients, which actually relates to, respectively, a 12-month median follow-up and an 18-month median follow-up, clearly show that we have trends that are dose-dependent, that produce an improvement in ejection fraction, a reduction in infarct size, a reduction in major adverse cardiac events, a reduction in all other cardiac events, and, at least until date, a really significant separation on mortality. We've still not had a death in the treatment group, and of course deaths continue to accumulate in the control group. So from all perspectives, from a clinical trial design this is, in fact, a very positive study and one that compels us to go forward in development.
David, thank you. Close with me now, because I agree with you that the phase 2 data actually in its totality looks very compelling, shows that there's a signal here. When you look at the global geographies and the global opportunity, there's been a lot of discussion about Japan. Mesoblast has been talking about kind of what they're doing with their pharma. Athersys just signed a new partnership in Japan. Is Japan a place that you're looking at?
We're more than looking at Japan. We have active dialogue going on, and just last week our scientific team was in Japan meeting with the Japanese authorities to finalize a phase 2 program in critical limb ischemia, which is a complement to the other cardiovascular program that we have, whereby we could take advantage of the new regenerative medicine laws in Japan. We've also had discussions in Japan on a number of our oncology programs, and it's clearly a place where not only do we have the opportunity to take advantage of the evolving regulatory climate, but that specifically to NeoStem, we have several members of management, I'm being one of them, that have had long ties to Japanese KOLs, the Japanese regulatory authorities, and Japanese companies. I was, in fact, the U.S. CEO of Chugai for many years. And so I think Japan will become a news item for NeoStem sometime in the next 6 months.
Very exciting. Not the same company where I worked a few years ago, how quickly things change. We ran out of time today to talk about the T regulatory program, but when I look at the valuation disparities that exist between the NeoStem program and what's happening in that space, I also see we could spend a lot of time talking about that. Thank you so much for joining us today.
Thank you, Jason, and thank you all.